Welcome, Tommy, to the Single Cell World podcast. And first, thank you so much for accepting my invitation. Okay. Second, I want to tell you that the title of your video can be transformed into one of the frequent questions that I get from single cell wet lab researchers. How can I transform myself into a computational biologist? And I would love to hear from you how you did it, because this is also one of my questions. How can I learn computational biology? But let's start from the beginning. Where are you from?
Okay, first of all, Katia, thanks very much for the invitation. Yeah, I'm from China, originally south part of China, a small town. And then I went to actually Shanghai, it's a pretty big city, to do my bachelor degree. And like I grew up in a low-income family. My dad was a teacher, middle school teacher, and my mom didn't have a job. She was caring for me and my sister at home. And when we were a little bit older, she started to actually sell some groceries in the school to make some money. And she also learned how to mend shoes. Actually, it's a really hard job. Actually, summer and in winter, and she has to be outside. So, but she always taught me, okay, Tommy, like if you want to change your destiny, you have to learn, you have to you have to study. So I studied really hard when I was small, when I was young.
Yeah. And when did you decide I want to be a scientist? It was when you were studying a lot, like you said.
Yeah. To be honest, I didn't really probably want to be a scientist, or I didn't. Let me put it this way: I didn't have the idea of what a scientist is and how it looks like. Maybe actually when I was in the 5th grade, I wanted to be maybe a businessman. I said, okay, so I can have some money, I can like improve the life of the family, And but then, like as I told you, like the life was hard, but I studied pretty hard as well. So I actually got into the best high school, the best middle school first, and the best high school. Like I was ranking number 1 in the county.
Wow.
Then I went up to Shanghai. Jiaotong University is also one of the top schools in China. So really there I had the opportunity to learn a little bit more about what's out there, because it's a good university and they have a really good atmosphere for students to go abroad. So they have organizations essentially teach you how to study the exam, because you have to take the TOEFL or like GRE, those exams, English exams, to be able to actually get into the US. So it was really good atmosphere there. So I started to prepare for those exams. I didn't know like I want to be a scientist, but then I mean, for me, the best thing for me to get a PhD actually in the States is I got a stipend. So it's free actually to do a PhD.
I know.
And also they give you the stipends. I think that's actually the primary reason that I got into science, and then I joined this wet lab to do a lot of molecular cancer biology study, like at the University of Florida. That was back in 2008.
Okay. Yeah, don't worry, because if you ask me also when you decided to be a scientist, I will tell you I didn't. In my case, it was like opportunities that were showing up, and I had, okay, I like this. Then I tried the wet lab. I like this. I will continue. It was a little bit like that because, as you said, I didn't know back then what was to be a scientist, right? So
yeah. Life is very interesting. Like you may think everything like happens in random, but then when you look back, oh, there's a path actually to lead you here.
Yes. Yeah, that's right. So the PhD opportunity came in the States, as you mentioned.
Right.
It was like in the studies that you were doing, you were getting prepared to do a PhD in the States. This was something that you saw as an opportunity, or was opportunity that came out from your studies?
So my major was plant biotechnology. So I was applying to anything that biology related PhD programs in the States. So I was just looking online, okay, which school has a PhD program in biology, like genetics. And then also I need to think about, okay, those top schools, maybe my GPA is not good enough. So like I actually applied like 15 or 20 schools. And even
Mm-hmm.
for this University of Florida, actually, I'm really grateful. Like I was on the waiting
Mm-hmm.
list. So really like the second round, I got into this program and I was
Yeah.
really lucky. So that's
Mm-hmm.
how I did that. So I applied a bunch of them, really top ones, middle ones, and then
And did you got
yeah,
a
yeah.
lot of rejections? Because this is something also that when people listen to us and they think, they are like in this position now, I don't know, they have a lot of followers. People think that we never kind of, or we were not rejected our
No,
grants, but it's not true. And this is really
um,
important to make it
yeah,
clear that I got a lot of rejections. What
yes,
about you?
I got a lot of rejections too. So I mean, that probably was the first time that I feel a little bit desperate because I was a really good
Yeah.
student. Like when I was in high school, I ranked number one. No one actually succeed actually overnight. But then, actually, if you see somebody succeed, that's 10 years work actually after
Yes.
before that, right? So of course, I got rejection from many schools. Even for this one, I was on the waiting list. But I was lucky enough to get into the program, and I'm really grateful for that because I don't have to pay for the tuition, and
Mm-hmm.
I get a stipend. I can support myself. I even actually saved some money and give it to my mom. So which
Yeah,
was
your family.
yeah.
Mm-hmm.
Yeah.
Yes, because when you move to the States, I saw in your YouTube video that I really recommend to everyone that is listening to us to go and check your YouTube video where you talk about your experience. So the movement from when you moved from Shanghai to the States, you didn't had a lot of like money to go with, right? You had a little bit a small budget that you needed to manage, I suppose, when you went to the States. Right?
Yes,
Yeah.
yes.
Yes, yes, yes. This is also important. Yeah, because of this, sometimes is that people our think lives, that, for example, of people of researchers that are now like the reference are very easy, right? Especially for the young people, but it's not. There are always um, challenges that we have to overcome, and
Yeah,
this
yeah,
is really
yeah.
important.
I was a single, so even
Mhm.
though like many people complain, okay, the PhD stipend is not maybe enough. I mean, of course, if you have family, it's kind
Yeah.
of like minimal. But I was single. I was sharing like rooms with other students,
Yeah.
so my cost was actually really low. So, but I actually just enjoyed the time and
Mhm.
to learn a lot of new things, the wet lab technologies, all these assays. I even did some like northern blot, really old technology back.
Yeah.
Yeah,
I
in those
also
days.
did and southern blood. Yeah, yeah.
Yeah.
I think maybe some people will not know what is this anymore.
That's true.
And when did you decide to share your experience, to share your knowledge on the social media?
Uh, really, okay, as I mentioned, like I was actually studying in a molecular cancer biology lab, wet lab, right?
Mhm.
And 4 years into my PhD, I already had two first order papers in respectable journals. I was pretty happy. But then one day,
Mhm.
my advisor asked me to analyze this public available chip sequencing data. So that's around 2012. And I downloaded the data. I just couldn't open it with Excel. It crashed it. So, and then the first time also, maybe the second time I realized, okay, maybe I need to learn some data analysis skills. So that's
Mhm.
actually when I started to share things on social media because I actually before that I read a blog post by Stephen Turner. At that time, he
Mhm.
was the bioinformatics core director at the University of Virginia. And he has a blog post, How to Stay Current on Bioinformatics. And at that time, I
Mhm.
just started to learn. And I'll say, oh, you have to follow these people on Twitter. And that's how I actually started my Twitter account to
Mhm.
follow people. And at the same time, he also suggests, okay, those are the blog posts, blogs that you need to follow. And then I started my own blog. And really, what I did initially was just to share what I learned, actually. Okay,
Mm-hmm.
I had this problem. I don't know how to install this tool. I don't know how to analyze this data. And I figured it out by Google around. Then I just wrote it down and in a blog. And then I shared it on Twitter with other people. Yeah.
Mm-hmm.
That's how I
Yeah.
started 10 years ago.
Oh my God, 10 years ago.
Yeah.
Yeah. The other day, your post on Twitter where you explained that 10 years already passed and you, at the beginning, you didn't have a lot of followers. To be honest, remind me because sometimes it's true that I think, I will never have so many followers as these. Everyone compares themselves.
Yeah. Yeah.
And when I saw your tweet and I thought, yeah, it's true, Katya. You need time. We need time. Yeah, yeah.
Yes.
It's
Yes.
time
I mean,
and yeah.
if you take the right perspective time,
Yeah.
you are set to succeed because as we all talked about, like no one succeed overnight, right? It's all
No.
about decades work, right? So when I started, I didn't have any followers. Like I started with zero, right? So, but then what you do, right? But of course, initially I was just tweeting random things
Yeah.
like complaining about the bioinformatics problems. But if you really want to increase your followers, I mean, there's a key thing that you have to follow. You want to add values to your audience.
Yes.
But
Mhm.
at first, you don't even have any audience, right? So what you need to do, you have to go to those big accounts. Like, for example,
Yeah.
they already have their audience. You comment in their threads. Okay, valuable, comments, ask genuine
Mhm.
questions, and their followers will see your comments and they will come to see your profile. And if you give them
Yeah.
values, they will follow you as well. That's how it started. Yeah.
Yes, yes, yes. No, I cannot complain. To be honest, I cannot complain. I didn't have zero like followers, but yeah. No,
Yes.
yes. It's question of time. As with everything, we need just to keep going, doing a good job, and not quitting. Anyway.
Yes.
So, what about single cell data analysis? Let's see. Is it so different the type of analysis that you do when you are comparing it to the bulk data
Mhm.
analysis of
Mhm.
RNA-seq?
Yeah. Okay, let me start with my own story, like how I started
Yes,
to analyze single cell data, or
sure.
even how I started to learn computation skills, right? So you also mentioned actually before this, like you have to master the basic skills first, right? Just like you are doing
Mm-hmm,
experiments, you know how to culture the cells, how to extract DNA, how to extract RNA, those things, right? For data analysis, you have to master those basic skills. For example, Unix, you know how to use command line because most of the bioinformatics tools, they're written to run on command line. Okay.
mm-hmm,
And second is you can learn Python or R, but then you have to learn how to wrangle the data, meaning how to uh, take in the data into different shapes, so or clean the data. Those are the basic skills, right? And after you master those basic skills, then you pick one of the like sequencing, for example, data type to start with. For me, I started with chip sequencing because I
mm-hmm,
was in the gene regulation lab and we study a lot of transcription factor binding or histone modification enrichment in the genome. Okay, you just take one data type from public data repository. You download the raw data and take a look how it looks like FASTQ files. What does that mean, right? So
mm-hmm,
it's
yeah,
from the sequencer, like FASTQ files.
yes.
And then you just go through the process, like the routine process. Okay, FASTQ files, I need to align to the genome. If it's
Mm-hmm,
RNA-seq, I need to map to the transcriptome. So there are different tools for those process. And then you should learn for one data type. And then you can
okay.
easily extend to other data types. For example, I didn't know how to analyze single cell data, but I started with
Mm-hmm,
ChIP-seq, then RNA-seq, then whole genome, whole exon, like
mm-hmm,
for mutation callings. Then I also did some bisulfite sequencing data, ATAC sequencing data.
yeah.
Okay, then it's really because I have mastered all those basic skills, like how to run commands, how
Mm-hmm,
to wrangle the data in Python or R. Then it's really a matter of getting to know the standard process for a new data type. For example, for single cell, because you already have the experience of bulk RNA sequencing, actually it's very
mm-hmm,
similar as well because
okay.
yeah, for bulk RNA sequencing, it's like you have multiple samples
Mm-hmm.
then you have 20,000 genes, right? But then for single cell, it's really 20,000 genes, then the column will be millions of cells or tens of millions of
Mm-hmm.
cells, right? But then the process is similar because for single cell, you also align those reads, also the FASTQ files, the same thing, align it
Yes.
to the transcriptome. But now the difference is that now you have a cell barcode, so you know this transcript is from which cell. So that's the only
Mm-hmm.
difference, right? And in terms of pre-processing. But then in terms of post-processing, in the end, as I mentioned, you get, so you from the raw FASTQ files, then you do the alignment and you also do the quantification. Essentially, you count how many reads is from this
Mm-hmm.
gene, from this cell, right? Then in the end, you get this huge data matrix. So the rows are the genes, 20,000 genes in
Yes.
human. Columns will be the cells. So essentially, for single cell, you start with that for your downstream analysis. And for bulk RNA sequencing, the rows are the genes, columns are the samples, and that's also the data matrix. That's why you really need to start with the basic, because essentially you're dealing with data metrics. So if you
Yeah.
know R or Python, how to subset the data, how to transform data, how to principal component analysis, skill set
Mm-hmm.
is the same for different data types. Of course, single cell has its own challenges because there are many dropouts, or people say dropouts, like many
I
zeros, like
know.
because it's very sparse. These are different terms, so because there are
Yes.
many zeros for a cell like certain gene. So and you have to treat it a little bit differently. But I would
Mm-hmm.
say in general, if you know the basics, you can always apply the skills to a new data type. That's how I learned. Yeah.
So right now, after you explain this, now I know that I know, like now I have more knowledge than I thought I had about data analysis. Because why I asked about a comparison between bulk and single cell
Mm-hmm.
data analysis, because I did bulk data analysis.
Mm-hmm.
But for example, I didn't realize that the basic skills, as you say, that I use there can be
Mm-hmm.
like translated for single cell data analysis.
Yeah.
That is good to know.
Mm-hmm.
So yeah. So the first thing that you will recommend to someone that is 100% wet lab
Mm-hmm.
person, but that person will know about the sequences, about the FASTQ. I
Yeah.
think is there where the knowledge stops when we are 100% wet lab. So what skills should we learn? Like the first skill should be code, right?
Yeah, so it really depends on like
Okay.
your interest, right? So
Mm-hmm.
if you like if you are not
Mm-hmm.
really into coding, probably don't force yourself to learn. Find a
Thank you.
really find a really good collaborator, right? So communicate with
Yes.
them. I think that's the key. But really, if you want to learn computational
Mm-hmm.
biology, like what I did, because I really like to do
Yes.
things on computers, so that's why I spent so much time during my PhD, like learning online and reading
Mm-hmm.
books, And if you really want to learn, I think the first thing you want to learn is Unix commands. I think
Okay.
it's everywhere because you have to know how to use it, because all
Mm-hmm.
the commands, like all the tools, for example, alignment, they are run on the commands like BWA, like star, like for those aligners, you have to use commands. So, but of course,
Mm-hmm.
the next thing that you want to learn is a language like Python or
Okay.
R. Like
Mm-hmm.
either one is okay, like depending on which one you like.
Mm-hmm.
Before, like for me, I pick up like R, and because it's just more intuitive to me. For more people, like Python is more
Okay.
intuitive. But I use both. But I primarily use R for data wrangling, data visualization, and the other good benefit of R is it has a really good community called bioconductor. So it has many packages that you can use to analyze different type of data. It's
Mm-hmm.
already built by others. So and the visualization is very nice in R if you know ggplot2, and
Mm-hmm.
highly recommend. So if you learn R, so you learn how to wrangle the data, shape the data, how to visualize the data, and that's most about that.
Mm-hmm.
Yeah. But if you really
Yes.
want to go deeper, like I want to apply machine learning, other stuff, then you can learn a little bit other stuff. Yeah.
Okay. Yeah, because like the advice that you gave to me is what I normally I say. Instead of learning, unless you really want to learn data analysis,
Mm-hmm.
instead of learning, do a collaboration with someone that is expert on data analysis,
Yeah.
specific for the type of data that you will get, because single cell RNA-seq will be differently analyzed when we compare to single cell attack-seq. So
Mm-hmm.
a person that really knows how to analyze the type of data that you have. This is always my advice. But in
Yes.
case someone will like to learn, so
Yes.
start with what you said. Yes,
Yes.
what
Yeah.
you mentioned. Yes.
Exactly. So you have, because if you are in a PhD program, you have to publish
Hmm.
this paper, right? And
Yeah.
it has a time frame. Like you have to do this, finish this like within
Yes.
2 or 3 years. And as I told you, everything takes effort. Great things takes
Hmm.
great effort. So you might be able to learn like how to analyze single cell data, but it can take you like several years.
Yes.
So you have to just be prepared for the time. Yeah.
And what is like, if someone, it is a person that really likes, it's into learning data analysis, how long it will take to get like the basic skills? Do you have an idea? I know that it will
Yeah.
depend with the person, but
Yeah.
how long
Yeah.
it took to you?
It took me like almost 1 year to get really comfortable on the command
Hmm.
line because it's very different from what I usually interact with the computer, right? So a command line
Hmm.
interface is just a black box.
I
And
know.
then there's a like cursor there blinking, waiting for you to type in things. You hit enter, and then the computer execute that program and give you some output, right? So you have to be really comfortable like with that and navigating in the system, go to the right folders that you want to go to. And you know what? In the very beginning, it was funny. Like I see people doing so fast, they're typing so fast. I was, oh, it's amazing. Like how can they do so fast? And later,
Hmm.
I learned that they're just changing into different like directories, like different folders. Like
Yes.
it's really fast. But because you are so used to it, because the memory is built into your muscle, your fingers, you can just do that really fast.
Yeah.
So it's all
Yes.
about
Yes. Yes.
practice, like
Yeah.
anything.
Anything. I have to be honest and tell you that the black box makes me afraid. It's like, what is this? It's like
Initially,
if I,
I was afraid of it too, but once you master it, you will
yeah,
love
I
it.
know, I know.
You will
Yeah.
love it. Yeah.
Yeah, I know, I know. It's, do you know, my feeling was like, okay, if I write something wrong, it will delete everything that I have in the computer. This is what I'm afraid of. No, no.
Yes.
It's
Yes.
what you said. It's practice, and as always, learning, learning. I know it's happening every day. We are with, I don't know, Chat GPT.
Hmm.
We are learning, at least me, I'm learning every day how to write the prompts
Yeah.
properly to get. But
Yes.
so yeah, as always, we need to make the effort to learn, and yeah,
Yeah.
and the time needs to
Yes.
pass.
Exactly.
Yeah. And now I have like a question, your opinion. Do you believe that the single cell company's software, it will be sufficient to analyze the data to publish? My opinion that it is not, but I'm not an expert on, on data analysis. What about your opinion?
Yeah, I think there are many nice actually software tools, like
Mhm.
commercial available tools that you can do certain analysis, and those like maybe GUI thing that you can point, drag, and click those things. I think I think for the regular analysis it can, but
Mhm.
my experience that you always need some customer analysis, which
Yeah.
the because the software is like a general use, like people can do the same thing, but then for each project, for each experiment, you have some specific questions you want to ask. Then you really need to actually be able to manipulate the data, subset data, dig into this, ask a different question to test different hypotheses. So I don't think it can actually, yeah.
Mhm.
But
I agree.
some of those tools are really useful. So what I did in my last paper when I was in Dana-Farber, so I was the computational biology. I was doing the analysis
Mhm.
purely on commands, and but uh, the web biologist also wants to play with the data somehow
Yeah.
interactively in the GUI, right? So 10x, they have their own browser, and we were using BioTuring, this browser, and it's really useful. So I can create thread object, if this is a package for analyzing single cell data in R, or ScanPy project object, I can give them to those web biologists. They can actually upload those into those interactive, software, and we are actually looking at the same thing. So actually give them really intuitive, actually figuring how the data look like. So then we can communicate with each other. They find something interesting. And if the software cannot do that analysis, they will tell me, okay, Tommy, I want to do this. This software cannot do it. Can you do it for me, like in R, in Python? Yeah.
Yeah, that's really cool. Yeah, there are a lot of softwares, not only that are commercialized by the single cell companies, because now what I see is that a company that sells a kit, single cell kit, has also software specific to analyze their data. But besides this, there are other companies that are more data analysis, and they also have a software that is very interactive and it's very easy, friendly use for wet lab researchers. Yes.
Yeah. There's a need, actually. Definitely there's a need there, because those web biologists do need a tool, software that is easy to use and for them to actually get to know the data quickly, right? So there's a need for that, definitely, yes.
Yeah. Now, for a wet lab person like me, so we are always a little bit afraid of these softwares not to be enough, right? And I don't know if you agree, but again, I always advise for someone that is expert on data analysis to be included in the projects. And this person should be included in the beginning before you start planning the experiment. Do you agree with this?
Actually, I was gonna say that. So you want to actually include a computational biologist or bioinformatician or even a statistician before you carry out the experiments, right? Because one of the mistakes people make is introducing a batch effect for your experiments, right? So if you have two, one is control cells, the one is the treatment cells, and if you just sequence them or do the experiment in different day for your control and for your experiment, for your like drug treated, for example, and you do them in different day and you sequence them in different day, then in the end, you really don't know the difference. It's because of your treatment or it's because those technical issues. So you want to have a more balanced experimental design, so you can have your treatment control and treated group sequence in the same day, and then you also have another batch sequence in the same day, and then you put them together. At least you can actually separate them by computational methods.
This is that you are telling is really extremely important. I will say it's crucial for us to have good results, good quality results. So some people, they don't really think about this, how to decrease all the ways that we can decrease batch effects while we are planning our experiments. This is completely crucial. So let me see more questions. Yes, you have a book. Well, you have different books, right? But there is one where, by the title, I didn't read it yet, but I will, that you explain basically how a wet lab researcher person can transform the skills, I will say, skills or get new skills and become a computational biologist.
Right.
Can you tell me a little bit about, like a sneak peek about this book that you have?
Sure. So the title of the book is From Cell Line to Command Line. Then essentially how I transform myself from a web biologist to a computational biologist. So really, because during the past 10, 11 years, I learned quite a bit. So I accumulated a lot of notes and small things that I learned. Also, I wrote a lot of blogs, posts. And what I really did is I kind of summarized all those things together into a book format. So to help you to learn with a clear roadmap. Because when I started, I really didn't know even how to start or where to start. Which course should I take? What books should I read? And in this book, I actually outline all those really useful resources that I collected. And also, I demonstrated in code how you can analyze some common data types, for example, bulk RNA sequencing. and how you can actually organize your bash scripts into like workflows using, for example, SnakeMake workflow languages, and how to deal with spreadsheet, how to use spreadsheet correctly, like use consistent names, like those small things. So, I mean, to be honest, like many people even don't know how to use spreadsheet appropriately.
Yeah.
And there are many tips there, like about 400 pages, like in PDF,
Hmm.
and yeah. So if you want to take a look, it's in my
Yes, I
website,
will.
diving into genetics and
Mhm.
genomics.com. That's my website. So you will find my book. Yeah.
Yeah, I will leave all the links in the podcast description for everyone that is listening to us to go there and have access and have a look on your blog, on your YouTube channel, on this book. I will read this book and then I will give you the feedback.
Sure.
No, yeah. No, it seems I love the title. I thought, here is a book that I will read for sure. And is there some other resources like, or for example, a lot of people ask me, do you know about a data analysis course where I can learn about single cell data analysis?
Mhm.
I'm always saying, no, I don't know, because normally I try to recommend the things that I tried, online courses. But do you have any recommendation? You are an expert on this?
Yeah. I think you can start with some data carpentry courses. Those are
Hmm.
free. So if you don't know what's data carpentry, you can just Google data
Yes.
carpentry, and
Hmm.
it's a nonprofit organization teaches like non-programmers how to do computation. So, and they have different curriculums. And one of the curriculum is called genomics. So it teaches you how to use command line, and how to use R to do, like, for example, RNA sequencing analysis. So those are the basics. As I mentioned, you want to master those basics first, right? And then uh, you for single cell specific tutorials, I like in my GitHub repository, I have one specific for single cell, and I collected many uh, open free courses that you can take from other
Hmm.
universities, and you can go there and take a look if you want to
Okay.
just do single cell. Yeah.
Okay. Yeah. And now my last question, and then is your free time. You can talk about whatever you want. Is what was the biggest challenge when you were doing these, like when you were kind of translating from the wet lab to the computational analysis?
Yeah, the biggest challenge, as I mentioned, is I didn't know how to start. Even I
Yeah.
didn't have people to turn to. Like when
Hmm.
I was at University of Florida, I was the only one on the floor because who was learning bioinformatics because it's a wet lab, and
Yeah.
and didn't have people to turn to. And I had a lot of questions, but I don't know how to solve. So I had
Hmm.
to actually Google around and go to those forums. Like, so those are two forums you also want to go to, like biostars.org, and the other one called
Hmm.
seekanswers.com. So those are bioinformatics forums that you can ask questions and people can
Hmm.
help you. But I also have a video how to ask good questions. So make sure when you ask questions, provide enough details, context, so people
Yeah.
can help you easily. So the rule is that you are asking other people to help you, and your job is to make their life easier, because you are already consuming other people's energy. You want to make their life a little bit easier to answer your question more easily. So
Yes.
provide enough context. Even paste some of the code that you wrote into those posts, so people can really diagnose the problem for you. Yeah.
Hmm.
And I would say, yeah, that's the biggest challenge. And for now, there's so many online courses that you can take. I think you just need to
Yeah.
find a course and really dig into it and learn the basic skills and start with a
Hmm.
new real project, so you can really practice what you learned theoretically
Yes.
into practical life. That's always the key, how you learn. Yes.
Yes. Yeah, nowadays I will say that there is so many information that is very difficult for someone, like it is happening in the wet lab. It is difficult for someone really know what is the best for them. Like, for example, I'm talking about kids. There are so many options that people say, okay, I want to do this, but I don't know what is the best.
Yeah.
And but for that, they have your Twitter account or X account now that they can follow.
Yes.
So follow Ming, Tang, Tommy on Twitter. You also have LinkedIn account,
Yes, I
right?
do. Yes.
Do you have other social media?
No, that's
Or
the two major ones that I'm using. Yes.
okay, yes. So yeah, and just as I always say, ask people, ask, for example, us, that we can reply to all the
Yes.
messages. And
Yes.
yes, that's so. Yeah, would you like to tell something else?
Yeah.
Some
Yeah.
comment?
So
Yeah.
I just want to encourage everybody who is at the early stage of your career, or you're at the early stage at learning something new. Just be persistent, and you 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 take the time, you take the efforts, and you will get the reward. So
Mhm.
don't think you can be like you can be an expert of data analysis in 3 months, right? So it takes years of experience. So take your time, just be present there, like, learning new things every day. You will see the results in 10 years. That's
Yeah,
my advice.
I completely agree. I always say, when people say, because you know so much about the technology, I always say, yes, but I have an advantage. I'm older. So a lot of time passed where I get all this knowledge of the years, right? My
Yeah,
experience, everything. So this to tell, as you said, when people are young and they compare themselves to us, yeah, it's impossible for them to know
right.
so much as we
Yeah,
do.
yeah, yeah.
So it
It's
will take time.
yeah, yeah. Time is the currency that you have to pay to accumulate
Yes,
experience.
yes.
Yes,
And also say to everyone that is listening to us, take advantage that like ourselves through social media, and do questions. If we don't know and we cannot help, we will send you to someone that can. Because I'm sure, I don't
exactly.
know about you, but I know a lot of people that, for example, in data analysis, that I don't know, but I say, okay, contact this person, ask this other one, and this, I think it's essential. So yeah, thank you so much for being here today, and see you next Monday. Thank
Yeah.
you.
Thank you,
Thank you
Katia,
so
for,
much.
for having me. Bye.
Thank you. Bye. Okay, I will stop recording and now thank you so much.